Good morning, folks. I hope everybody's having a wonderful time at AbSciCon. It's good to see a nice crowd here at 8:30 for our plenary session. Before we get going, I would like to remind everyone of what promises to be a really fun event tonight, entitled When Worlds Collide. Many of us are aware from personal experience how science and the arts grow from the same roots and reinforce one another and mutually inform one another. And so this issue will be explored this evening, and there will be a self-described geek musician, Mr. Griffin, who will be performing, and I think you'll all get a big kick out of that. So please join us at 7:30 here in the same venue. So it's my pleasure to start this morning's plenary session and give an introduction to today's speaker, Professor Betül Kaçar. So a long time ago in a galaxy far away called Montana, I received a message from Michael New. And specifically, Michael summoned me to AbGradCon, which was taking place in the fly fishing capital of Ennis, Montana. Specifically, Michael summoned me, and so having just received an exobiology grant, I immediately hopped into my old Land Cruiser and drove the 3 hours from Missoula. Lucky for me, the drive was, as always, spectacular, but also the interaction with that young scientist was delightful, and it was auspicious as well. And so over the course of more than a decade, it has been my pleasure as well as my privilege to witness the growth and maturation of today's plenary speaker. Dr. Kachar received her BS from Marmara University in Istanbul, and then went on to earn her doctorate in biological chemistry at Emory University. She pursued postdoctoral studies at my home institution, Georgia Tech, where she began what I would call her scientific vision quest that she'll share with us today. But what do I mean by that? Well, I would direct your attention to a 2011 paper in the journal Artificial Life, entitled Towards the Recapitulation of Ancient History in the Laboratory. This was what was on Dr. Kachar's mind when we conversed in Ennis. Now, at that time, ancestral state reconstruction was a well-established practice in phylogenetics. What was daring about Betül was that she aspired to carry on ancestral state reconstruction in the laboratory using integrated synthetic biology and experimental evolution. So did she realize these aspirations? I would direct your attention to last month's article in Cell entitled Resurrected Rubisco Suggests Uniform Carbon Isotope Signatures Over Geologic Time, a real masterpiece integrating all of these different fields with geology. Now, I would like to emphasize to the young investigators gathered with us this morning that the path that led from Istanbul to Ennis, Montana, to Madison, Wisconsin, where she's currently a professor, was not an easy one. But along that path, Betül held fast to her vision and showed true grit, taking on challenges and overcoming obstacles and adversity that would have defeated a lesser human being. That, to my mind, more than anything else, is what makes Professor Kachar's journey, her story as a scientist and as a woman, so compelling. After Georgia Tech, Betül continued postdoctoral work with Dan Anderson at Uppsala University and Rich Linsky at Michigan State. And then she spent 3 years at Harvard, working with Scott Edwards before accepting her first faculty appointment just a few years ago at the University of Arizona. This past year, her burgeoning laboratory group moved from Tucson to Madison, where she is a professor in the Department of Bacteriology. Dr. Kachar has been the recipient of numerous accolades, most recently the Stanley L. Miller Award from the International Society for the Study of the Origin of Life, a Scilog Fellowship from the Simons Foundation, a NASA Early Career Award. Her laboratory in Madison is currently supported by a NASA Templeton Foundation grant, the Human Frontier Science Program, and the NASA ICAR grant that serves as a cornerstone for the new research coordination network, Life from Early Cells to Multicellularity. Please join me in giving me a warm AbSciCon welcome to one of our own, Betül Kaçar.
Wow. Thank you so much, Frank. It really feels like homecoming being here. I always define my job as one of the most special jobs one can ever have, to be a biologist on the only planet that you can be one, right? You can be a physicist or you can study physics or chemistry and geology anywhere else in the universe, but this is the only place where we can be biologists. That's, to me, amazing. And of course, this is the crowd that's trying to change that, so we can be biologists on different planets. And we know that the challenge is big. What if I told you that these two images, wow, it kind of feels weird to not have slides here, there. All right, one of these images is lifeless, whereas the other one is living. At the very superficial level, life can trick us for its existence. The image on the left, in fact, is lifeless. The fern that may be tricking you to be a fern is a silver dendrite. This is an outcome of a malfunctioning engineering experiment. And on the right, you're looking at a forest, a fern, a lot of life. So while at the superficial level, life can trick us, and I think this very aspect makes our jobs as astrobiologists very difficult, things start to fall apart the moment we go up a scale or down a scale and ask deeper questions about the image that we are examining. What I mean by that is that if you travel up a level or down, you have to experience, or what you are looking needs to express a level of complexity, or an increasing complexity or a decreasing complexity, which is what we generally think a living organism or a living state attributes. So let's just start with an organism. We have, if you go up a level, we have populations that form communities, that form ecosystems, they impact the ecosystem and ultimately shape the biosphere. If you go down a level, we have cell groups and cell. At the very bottom, we have molecules, and you can also strip them apart. We know that there are self-similar patterns of interactions between each layers, and I'm showing you B because I think they're fascinating as a very top-level example of what life, how it organizes itself. There are rules between levels, and there is different level of complexity in each layer. So let's just drill down this image a little bit. At the top, we have biogeochemical cycles, catalyzed or driven or moderated by populations. And again, if you go down a level, we have organisms and cells and subcellular micromolecules that compose cells. And finally, at the very bottom, we have energy input and substrates. And I know that this group is studying this aspect of life at various levels. So let's consider a modern ecosystem. And I'm showing an image of the dinosaurs, and I know this is tricky because when I talk about early life, at least when I teach at the university, most students think that I'm talking about dinosaurs because they're ancient, right? But of course, this group knows that dinosaurs existed in a relatively modern ecosystem. And what we care about when an astrobiologist looks at an image like this What will really get their interest is that it's not going to be the individuals per se, but the effect of the individual on the planet scale reservations. Exposed surface, atmosphere, maybe temperature. We are interested in understanding the impact of whatever the living state is in that very body with its environment. And of course, the challenge might then be to detect these impacts. And I'm not even talking about the detectability problem just yet, but we know that direct observation is not easy. Of course, if we find dinosaurs on a different planet, in our solar system at least, that will be easy to detect. So biologists are well aware of these issues and have amazing tools to study these interactions. But what biologists are lacking, especially molecular studies, I think, is how are the concept of how environments and evolutionary innovations impacted our planet. We lack an understanding of the underlying variation and the impact of this variation at the planetary scale over long time scales. We also don't understand the circumstances and timing of events that made the early Earth a much different place. And we know these things through various studies that come from paleontology, paleobiology, geology, geobiology, geochemistry, so on and so forth. So let's review what I'm talking about. Of course, we are interested in the birth of life, how this planet gave birth to this magnificent thing that we get to study right now. But in some cases, if you think about our Archean Earth or the Hadean Earth, it's really the best of times, but also the worst of times to study. It's the best of times because life emerged. It's the worst of times because we have very little to work with when it comes to studying the past. There is very limited record. It comes from very limited data that represents a very limited subfraction of whatever the living organism or the population was residing in that very locality that we are studying. And the bigger the mystery, the fewer the data. So let's review what these major innovations are over time. We have cellular life emerging around 3.8, say 4 billion. I put these arrows as absolute numbers, but of course they do not represent an absolute day. Emergence of cyanobacteria, eukaryotes, followed by green algae. And of course, these are underlined by, in this case, the second symbiosis event that gave rise to these, what I'm sharing here. And then we have animals and land plants and surface ecosystems. So when we look at the history of life on Earth, these are the top innovations, I think, that really impacted what we see around us today. This is magnificent to me. And if you think about it, all of these events are interconnected. It all comes back to the cellular life and some innovation that happened at that point that got transferred. I'm using the word transfer with a lot of conservation here. Maybe in some cases adapted, or it was a fluke accident, we don't know. But they all rely on each other. So if I am here and talking to you, it is because some innovation at the molecular level happened billions of years ago. The fact that we are having this very conversation relies on these interactions of the past. And some of these, in fact, majority of them are what we like to think of as singular events. They happened once, and nothing like them happened ever again. And we cannot compare them to an independent data set. We don't have any other planet with life, so this is what we have. Even in this place, we don't have an independent record yet, at least, to compare these events to. And they happened billions of years ago. You cannot have an ecosystem or biosphere without cells. I just recently read a paper that animals and the rise of animals may be due to evolution of nitrogen fixation and the impact of that with the rise of oxygen on this planet. So these are all interconnected, and it's quite remarkable that these events happened as singular themselves. And early life, it's complicated. If you were a bacteria looking for a date with a really, really old organism and go to your Tinder profile or just check it out, this is what you would see. It's some sort of, as a biologist who is studying biochemical and chemical and molecular systems, when I first saw this, I thought, this is it. So when we look at past, this is what we see. So the favorite food is complicated, relationship status is very complicated, age, it's very complicated, family name, let's not even go there. So this is what we have as an early Precambrian microbe. And the foundations of these jumps, when I call them jumps that I just showed you, that define our planet are completely unknown. And I will add another layer to this challenge. It is not even clear whether these events themselves were inevitable. Ta-da, right? So that's another challenge we got there. So let's revisit the image I just showed you, this time with some pretty pictures. But what we know comes from maybe some chemical isotopic probing, and those, if we are lucky, we have some fossil data. Again, as we go back in time, we have scattered information, and the record gets spottier and spottier. But I want you to look at the gaps between these major biological innovations in the history of life. And by gaps, I mean the timing, right? Hundreds, millions, in some cases, billions. And I will tell you now why, as a biologist, I find that amazing. Because we know that at the molecular scale, variation happens very rapidly, okay? We do not have an obvious way, deterministic or mechanistic, to link variability on the scale of minutes with microevolutionary innovation that expresses themselves on the scale of billions of years. Think about translation machinery. It's the computer of life, right? It processes everything that in every organism that ever existed on this planet has this computer in itself. It is able to generate a peptide chain, in some cases, less than a second. That is very fast. We know when we study molecule and molecular life that the variation exists in that very rapid speed. So how is it possible that a variation that happens at that really fast pace leads to these major innovations that happen every other billion year? I think this is a miracle, but also a curse at the same time. That's why I think studying early life is very fascinating, but also very challenging, because we don't have ways to link these two events. And I think the fact that we are struggling to even comprehend these things, or at least I do, also perhaps indicates that we do not teach biology and biological concepts the way we should. We don't learn them in the way we should. We need to integrate life's history when we teach life. And I don't think we do that, at least comprehensively at the moment in biology. So, as I mentioned, just adding another layer to the challenge, we have this issue of inevitability. And I think, in my mind, contingency is it's kind of a reasonable assumption to make here. Just because it happened that way doesn't mean it had to happen that way, right? So the case for this, especially in deep time evolution of these earliest organisms and biomolecules, is a pretty reasonable one. And we are aware of this because of the studies that come from chemistry and physics, that they provide us this awe of the chemical space that is possible in the universe. And then when you think of it, about the innovations on this planet, they each are sort of universe of possibilities themselves. So we know this vast chemical space, and we know how narrow life reduced it down to some selective amino acids. We know there's a handful of amino acids that life selected, or certain backbones that life likes to use. If you think about it, considering all the possibilities, it's a pretty selected menu, very limited. So while physics and chemistry can give us this awe and wonder of the general, what we need is to understand the depths of the particulars of life. In this regard, there is no substitute for fundamental biology in the assessment of planetary scale life in the universe. We need to understand these rules of life that enabled the selection, the subset of the chemicals that it studies and utilizes frequently. We need to understand these events on their own terms and with what little data we have available. And this is also coming without the benefit, and of course, maybe in some cases, the limitation of comparing them to any other evolutionary history. This was really where my focus has been drawn into, and this is really the crowd that made me think about these things and showed me these gaps in our understanding of life. And I've been thinking, how do we do this? How do we draw the benefit of all the tools and systems that we now develop to questions such as this? And it was inevitable that we need to develop systems, however limited that might be, and I would love to talk to you about that more, and we will be, to travel backwards from present. That let's develop systems where we can at least make an attempt to understand the hows. How can a molecular system give birth to such major innovation in its own right? I narrowed my focus to not only organisms and cells, but the proteins that enable majority of the geochemical cycles and metabolisms that whose outputs we get to study in the rock record. And I spent quite a bit of time trying to link what sort of protein system can completely realize not only the history of life, but enable us to study the impact at the planetary scale. It seems like a big stretch, even though I showed you how a cell, if you go up a layer, is connected to what we study at the planetary scale. Overall, the approach, in my mind at least, was quite straightforward. NASA loves to use the term, not a fruit salad, but we're making a smoothie here. And I always thought, well, that's great, but you have to be very careful with what you put in the blender when you make a smoothie, because if you use a wrong ingredient, you can ruin the entire smoothie, and there's no going back. So with this dramatic pursuit, I thought about, all right, so we have the genes and proteins, and yes, we have phylogenetic approaches that allow us to study their evolution today. We know so much by studying viral evolution, predicting where mutations might be to some degree. Yes, they have their restrictions, but let's try to stretch this at the deep time scale. And can we engineer systems with these phylogenetically reconstructed genes to reprogram the behavior of the cell, which we can then study geochemically in the lab? With that, started this pursuit of evolutionary reconstruction of ancient enzymes. Yes, the ancestral state reconstruction was frequently applied to understand the evolutionary mechanisms and particularly how mutations drive adaptation, but wasn't being stretched, I think, deep enough to understand the planetary scale phenomena. And of course, those applications, I was exposed to those possibilities here. And here's a paper by Margaret Dayhoff, published in 1981. This might be one of the papers that she has that have the fewest citations, maybe 20, 30. We might be half of those citations. This is a remarkable paper, Evolution of Major Metabolic Innovations in the Precambrian. Margaret Dayhoff was a biophysicist. She revolutionized how we study how we utilize computational power to study protein structures. In this paper, they examine pathways where there is information on the chemical structure of enzymes. So they focus on extracting some information from the structure of the proteins. And they assume that the gene products whose structures have been conserved of these proteins have remained relatively unchanged in their basic functions over throughout Precambrian. So there is this assumption in their paper. It's a very brief paper. I highly recommend that you read it. We make this assumption very frequently, especially when it comes to understanding early life. We, in some cases, have to, but we know that it's not that straightforward when it comes to understanding how mutations and variation impacts protein structure and how that consequently impacts protein function. Single insertions or deletions can change the behavior of a protein altogether. We now know that it's not all about the active site, meaning the heart of the protein where the reaction, whatever the specific function happens. We know they operate in a complex network inside the system, inside the cell. And a single mutation that changes the structure of a protein can impact the entire cellular function because of this disturbance in the network. A single insertion can be as dramatic as from macron to macaron. It's true. with that in hindsight, we worked towards establishing a method. The idea was to start with protein sequence alignments, form phylogenies. We spent a lot of time on this, and I will show an example how we utilized our assessment of protein tree topology and our alignment output. So I will be drilling down a little bit there towards the end. Well, we inferred ancestral sequence. This is where the synthetic biology part kicks in. We can synthesize these genes. It's getting cheaper every year to create these things. It's not cheap, but it's cheaper. And in one level, we purify these proteins to look at their behavior in vitro outside of the cell environment. But what I really was interested in is, again, change the behavior of the organism to the degree that we can then implement biogeochemical applications to interpret or at least provide, who knows, an entirely independent data set using artificial biology in the lab to see what life can do. We started, the translation was my first focus. I started studying how this machinery that I just referred to you elongates peptides. These are very, not only they are singular in their own right? There's only one translation machinery. It may be different. If you look at bacteria or archaea and eukaryotes, there might be differences, but there is only one computation center inside us, inside all of us. So I started by poking with the elongation step of this translation machinery inside bacteria. I was curious to see what will it do to the entire cell. Well, when you poke with something that is really essential, turns out it breaks. So this was a very childlike, I think, venture. You know how kids like to break things. So we were just breaking cells all the time. But I was interested in understanding how far can the cell tolerate the perturbance that we are introducing by poking and by introducing ancient genes that are older and older. And translation is one of the first innovations of life anyway that dates at about 4 billion years, right? So we're messing with a 4 billion-year-old machine by asking it to interact and recognize a 3 billion-year-old partner. After spending some time working on this system and then subjecting these perturbed systems to evolution to see how they will repeat themselves, and I was interested in this replaying tape of life phenomena introduced by Gold at that time, I recognized that we cannot directly link such engineering to the environment. You can break translation in very different ways that may have nothing to do with the past. So we need to focus on proteins that actually interact with the environment in a bit more direct way than the translation. So while we are continuing that work for different purposes, we moved on because I developed an experimental framework for reconstructing these metabolisms. I thought, okay, here is what I learned. We need to have a geologic history that we are tying the behavior of these engineered organisms to. Is there a fossil or a geochemical record related to this protein function? There's a vast amount of literature coming from biogeochemistry on these topics. Do we have a phenotype, a characterizable and geologically relevant phenotype that we can generate in the lab? What are we linking this to? Is there an ancestry? Are we dealing with a tractable system? How far can we extend it to in deep time? Is there an appropriate modern host biological system? Yes, we can engineer a spinach, that's not a problem, but that doesn't tell you much if you're interested in arcane innovations. Do we have those model systems that will allow us to study deep past? With that, we turned into many different venues. One of this that I'm going to focus on is our way connecting protein evolution to metal availabilities of Earth over geologic time. There are some authors that contributed to these papers that are already in this room, and this is really remarkable, I think, generation of what astrobiologists also created over decades, that we understand to some degree that the metal concentration over geologic time did not remain constant for majority of these metals. We know this for iron, that has a higher concentration in the Archean, then there's a drop. And there's a lot of work into why such changes happened, the links of these changes to great oxidation event or different Earth surface-related phenomena. So this was very interesting to look at. And I can tell you, interacting with a lot of metalloenzyme biochemists, when I show them this image, they're mind blown. They tell me they never thought about history of where these metals came from. And I get it. But that's what we do as astrobiologists, right? And that's amazing that we are able to connect these seemingly distant fields together. Nitrogenase is one of these enzymes, a molybdenum-dependent nitrogen-fixing enzyme that also represents a singularity. It has a FeMo, the iron molybdenum cofactor. There's different versions of this enzyme, but this is the most prevalent version that exists today that fixes the otherwise inert nitrogen in the atmosphere. I think it is very amazing that life came up with this innovation once and has one protein to rely on to fix the nitrogen in the atmosphere. At least so far, that seems to be the case. It's not very creative. It is really putting all your eggs in one basket. So we rely on this enzyme to do its job well. We looked at the literature to study the previous studies that try to probe the evolution of nitrogenase over deep time. And not surprisingly, this came from astrobiologists here. Yet we don't quite know the exact timing of nitrogen fixation. There's a big question mark there, but in the origin of nitrogenases, we think must have coincided with the emergence of nitrogen fixation because that's what we see now. And that makes sense at the top level as a starting point. Our questions were as follows. What was the metal dependence of ancestral enzymes? Where did this enzyme come from? How did it evolve? And big shout out to Dr. Amanda Garcia, who joined my lab. I was a first-year faculty member at the University of Arizona 2018. And that's when she joined my lab as an NASA postdoctoral fellow to take this journey. Having been trained in paleobiology, this was amazing for me to have someone coming directly from the field trained in paleontology at UCLA to join our group to study molecular evolution system. It was exactly the kind of scholars that I was hoping to attract. Amanda asked the question, can we create phylogenies? And now, on top of building on the previous studies, a lot of these work came from Montana State. Can we use the updated sequence information in the database, rebuild the tree, and not only read the tree that we created, but also reconstruct the ancestors using this tree to understand the sequences and structures and the metal preference of this enzyme. She built machine learning models. This was a computational study that were trained on the active site amino acids of all the nitrogenases that exist today. Because not all of them prefer molybdenum. We have others such as vanadium preferring nitrogenases to come up with a classification to determine whether the ancestral metal dependence focuses on the molybdenum or not. We published this paper as our what the output of these analyses indicate that ancestral nitrogenases were in fact molybdenum preferring, which wasn't in contradiction with what was previously thought. We moved on to our second question, where did this enzyme come from? Again, we just because nitrogen fixation or something like it existed and that we read these signatures in the rock record doesn't mean nitrogenase as we know today existed back then. Don't we love to talk about life as we don't know it? This is an opportunity for us to explore a potential life as we don't know. We should not make these assumptions. So we asked the question, where did this enzyme come from? Did early nitrogenase was in fact a nitrogenase? For this, we looked at the partner in crime for nitrogenase, which is matrase. The protein network that enables this enzyme to have a metal factor requires maturases. As the name goes, the mature, they sort of, you can imagine them as maturing the metal for the nitrogens. They're sort of feeding them, put in very blunt terms. So we looked at their partners. Sometimes what you're looking for is right under your nose. And the person, or in this case, the protein that is doing the most amount of the majority of the work may not get the credit. So nitrogenases, we find, through creating a tree, a joint tree of nitrogenases and maturases, we did a lot of tree reconstructions, made different models. We submitted this paper to the Society of the Molecular Evolution. We wanted to make sure that we get the best reviewer feedback we can get, and we did. It was a great experience. After we reconstructed the ancestor and studying it, we found out that the ancestor was more like the maturase and not nitrogenase. So they may have evolved from their buddy, meaning the ancestor wasn't perhaps even fixing nitrogen the way today's nitrogenases are doing. These are all models, so I cannot assert these outcomes just yet. But if this is true, what might be the case then is that the molecular architecture required for the birth of this nitrogenase enzyme was unlikely shaped by the same environmental drivers that are implicated in the evolution of nitrogen fixation. So this is not a judging the book by its cover situation. Just because you have certain elements or metal in the environment doesn't mean life adopted that just because it was in the environment. There's definitely, we have to consider evolutionary forces and the laws of biology when we interpret the timing and the origins of such events. We moved on to our third question. All right, so how did this evolve? It's great to do computational work, but we have to get into the lab. So we rolled up our sleeves and back to our paleophenotype assessment methodology. We have the reconstructed ancestor. Let's engineer this inside a modern microbe. And let's be calm here. We have to work with an organism that is well studied because we don't want to reinvent an entire engineering pipeline for a new organism. And we are very grateful that a lot of amazing scientists are doing that. Let's engineer this ancient DNA inside a modern organism and then study what these ancestors will do. We picked azotobacteria. This is the prime nitrogen-fixing bacteria. It's a huge focus for a lot of studies that focus on, it even goes to climate change implications and crop engineering, and plant biologists love this enzyme. But yet again, and due to these, there was a lot of development in the engineering of this organism at the genomic level, primarily developed by Dennis Dean at Virginia Tech. So we visited his lab to learn these skills. We brought them back to the lab. We tweaked them for our own purposes. So we did the following. You are looking at the purple and the blue part of the screen. Just focus on that for now. What we did is that nitrogenase is encoded by 3 different genes, NIF, H, D, K. At least these are the ones that we focused for this study. We wanted to know whether we can swap the entire genomic architecture with the ancestral DNA, or whether we should follow a stepwise modularity. So we have to do these things in order to set the system up, because we have to understand the limits of what we're dealing with too. And of course, the majority of the question is that how ancient is this? It is going to be as ancient as we can make it at this point. So on one level, Amanda engineered the NIF-D with its ancestor. That's one gene out of the cluster. And then in parallel, she engineered the entire cluster with the entire ancient gene coming from the tree. And I'm pointing out where this is coming from. We are not yet at the deepest part of the tree. We are not yet at the deep Archean, not even there. We are still within roughly a billion year, maybe, if I'm generous. So we are not traveling deep time yet. I mean, a billion is pretty long time. So we want to test the effects of the ancestor H and the ancestral subunits. And I'm very happy to share these results. I have actually just posted this. It will be online today. So these are the freshest results that you're seeing. It's my first time showing them. That we resurrected these nitrogenases inside modern bacteria. You're looking at the growth curve, the optical density plot on the y-axis, and towards time, we're basically assessing the growth of these bugs. And I must say that one thing we have in universities are these amazing undergraduates. Big shout out to Arizona Space Program and Brooke Carruthers, who spent her entire freshman and sophomore year optimizing a growth curve plot. She's just fantastic, and she did this. We tried batch cultures, we tried test tubes, we tried multi-well plates, and what worked was the oldest, most traditional method is what ended up working. So we followed the growth of these engineered strains. It worked fine because that's how long it took us to engineer these strains anyway. So by the time we perfected or at least optimized the growth assessment, our engineered strains were ready. On the black line, you're looking at the wild type. That's the sort of modern, native, unengineered, unperturbed bacteria. Delta represents deletion. We delete one of these genes out of the chromosome just to make sure, as a control. And all the other blue and purple lines represent the strains with the ancestral nitrogenases. After we engineered these strains, we've also looked at the relative organismal activity and by looking at the acetylene to ethylene reduction. So we are still assessing the behavior of the entire organism. And you're looking at the reduction rate on the y-axis. We normalize this to total protein and different strains on the x-axis, and this was performed by Alex Riviere, and together with, of course, Amanda Garcia and Azul Pinochet Barros. So you might be seeing what I see, that these bacteria that contain the ancient DNA are active, and they are not vast differences, at least based on our experience, since we've done these with other systems as well. Fairly comparable reduction rates compared to the modern. We didn't stop there, and just a quick shout out, Amanda will be talking about this tomorrow in detail. As I said, these were still inside the organism, right? So we are still inside the genome, and yet, by the way, we do sequence the entire genome to make sure we don't have any other wild-type fragments left anywhere, that we ensure that there's no other modern nitrogenase DNA that is remaining in the system to give us any false positives. Moving on, we wanted to understand what does this enzyme, what would it do outside of the cell environment? So we extracted this protein from Azotobacter after overexpressing it, and then ran this through purifier column. And this was done by Derek Harris, also a remarkable postdoc who spent a lot of time purifying these ancient variants. Followed by enzymatic kinetics to understand the catalytic function of these enzymes. You're looking at the nitrogen reduction rates, and different gas headspaces were used. And Derek has published a paper previously to assess a mechanism to understand the modern nitrogenase mechanisms, and that the reductive elimination mechanism for the nitrogen binding being the core sort of characteristics for modern nitrogenases. What he is finding is that the efficiency is similar at the catalytic level to modern nitrogenases as well. And you're looking at the structures. These are the ratios for the mechanism output, and that gives us a clue about the catalytic efficiency of the extant molybdenum nitrogenases compared to the ancestral ones. So it's pretty good. And I've been working with nitrogenase experts. I am now in a department where these enzymes were even discovered, and azotobacteria was discovered as an organism. And they tell me that it's very easy to break these enzymes, or at least they're not as hard as I think. So the fact that we have, in some cases, up to 20–25% difference in amino acid, and we are still able to recover activity is quite interesting. Just to wrap up this part, the ancient environmental conditions do not fully explain the timing of critical and singular biogeochemical innovations alone, at least. And these experimental studies, I think, will be very critical to understand the molecular innovations of life's earliest traces. And we have done some work, as Frank mentioned, where we resurrected Rubisco, that is also thought to be one of those major enzymes impacting carbon isotope fractionation record throughout Earth's history. And because that work is published a couple weeks ago, I opted to present the latest one. So please check that out. But moving on, where are we going with this? Of course, this is all laboratory, artificial life. There's a lot of molecular component here. And keep in mind that integration of evolution with biochemistry itself is still pretty new grounds for biochemists. That we miss so much opportunity when we don't understand where life came from. Not just for astrobiologists, but when we study what we have, it's true that we have to understand the past to make sense of the present. We must do this, and we should not miss this opportunity to harvest all the sequence space and wildlife, and understand why life narrowed it down to a subset of what we study today. But this is where Muse comes in, because my desire was to connect these things to actual Earth, real life, to sites where people go and study the analogs for early Earth environments, where they culture these microbes, if possible, and study them. So that was my desire to work alongside with geologists. And this is very important, I think, for astrobiology, because when we assess the habitability or really when we analyze different data that's coming from different parts or wherever we are visiting, we are focusing on a problem that is existing largely in an organic chemistry space. But we need to integrate an understanding of the elemental composition and how such composition may impact habitability as well. So we are looking and working towards finding ways to transform this into a problem that can be studied in inorganic chemistry space as well. And perhaps you might be wondering, this is great, and what you're studying is at the time scale that is relevant for Earth, but how do we make this an observation problem? How can I, as an astronomer, study this? What is the use, say, for exoplanets or different observation? So in the remainder of time, quickly, I want to share one of our latest papers. We thought about this. We are largely at the geochemistry and evolutionary biochemistry space, and the blue and the green circles, but what we need is a more integration of the stellar atmospheric chemistry. So the idea was then, can we find molecules, proteins, that not only impact the Earth and fill in all the criteria that I just presented, but also may be of use throughout integration of, say, surface irradiance studies, or more generally, observation of gases biosignature level assessment. So with that, we turned into pigments. Our pursuit was to tie together bioinformatic analysis, geochemical and solar spectral models, atmospheric attenuation, in particular here radiative transfer calculations, and enzymatic observation. Simple. Characterize the coupling between the internal, this could be as internal as the inside in cellular or population, and external conditions, and infer biosignature characteristics generated by enzymes that have changed over time. One of the first enzymes we focused is rhodopsin. These are interesting. They're pigment. They're really actually fascinating. They look simple, but they're not simpler. They are tuned to the peak photon frequencies generated by the sun. And this is really fascinating, especially for biologists, I think, that when I'm from Black Sea, so I was always fascinated by the depths of Black Sea and how organisms deep that, and by the way, it really does look black if you haven't visited it. It's just remarkable that these microbes, what they can do to harvest the limited of the environmental energy that they got, and they come up with the most innovative ways to make use of the harsh conditions that they reside in. A phenomenon that I think most of us in this room can relate to, or some of us. So we have to come up with these innovative ways to survive. Life does this. So here is rhodopsin, Catherine Cephas. This was a perfect student. She was an undergrad at the astronomy department who also loved biology. So she came to me saying, I always want to be a biologist, but I also always want to be an astronomer. I said, well, I have the perfect project for you. So what she started with was to create phylogeny of these rhodopsin proteins. Again, we have this amazing amount of information that is in the literature, this vast amount of sequence data that constantly keeps coming from these wondrous metagenomic studies every year. It's really difficult to keep up with, which is very exciting. She updated a rhodopsin tree, created it. Here you're looking at different topologies. All these letters on the top refer to a different evolutionary model that we try to understand the best fit of our topology. So we go through this iteration. It takes us months. We want to understand, by studying the sequence, again, the ancestral functionality. What did this enzyme do? Can we relate to any modern function, to the past? We know that there is a proton pump function or chloride pump function for this enzyme today, but what did happen in the past? So after going through these inferences, we benefited from these two studies that came from University of Tokyo, where they developed back-to-back, they published these papers, machine learning algorithms to predict the wavelength spectra of the rhodopsin protein specifically. And they tested these the accuracies of these training models in multiple ways, not only through experiments that they do in the biochemical level, but also they generated multiple libraries, and they had a lot of experience in this studies anyway, and collaborated with marine biologists to test the accuracy of the predictions of their machine learning algorithm, which was also great because due to COVID shutdown, we had limited access to lab anyway. So really thankful for any computational researcher who makes their resources available in open access. And they were able to answer all of our questions throughout this process. Doctoral student Evrim Fer, she took this method and she wanted to understand what sort of different libraries we can generate for our rhodopsins to implement this machine learning methodology. So we weren't only interested in a handful of ancestors. What we can do computationally that we can't do in the lab is to try all kinds of variants. We can do thousands at once. It's really remarkable. So she created alternative sequences for every ancestor that we generate. We assigned a threshold for what we define as our confidence level, and also a threshold for the least probabilistic or most probabilistic ancestor that we created. And we identified the sites in the protein that are actually functionally important and generated these libraries and performed wavelength prediction. And through the suggestion of an excellent reviewer, we also tested the robustness of the machine learning algorithm to the training data as well in parallel. And for that, we created the wavelength prediction values for all these rhodopsin proteins in our tree that we created using archaeal and bacterial sequences. What we find is that the spectral properties of these ancestral rhodopsins indicate a green shift as we go further back in time, that the lambda max is in a range between 480 and 585. So what does this mean? Now that we have an ancestor and we can detect its wavelength spectra, we went to our friends at Nexus. And kudos to these research coordination networks, and this is really why they are there, so that you have your 1-800 astronomer that you can go and talk to and perhaps collaborate with to make sense of the biological data that you're creating. So Eddie Schwiderman from UC Riverside came to the rescue, having worked on this with Shildas Sarma for a long time. He modeled the Archean environment surface irradiance profile independently, and then we compared the data that we generate with the environmental, the surface conditions of early Earth. What we think is happening is that the green wavelength absorption is parsimonious with the spectral features of the early Earth environment. So we're looking for a proton pumping protein that may have residing in a microbe that is swimming in the shallow waters, or perhaps was underneath some biofilm. to shallow coastal marine niches, which is maybe a great environment, abundant nutrients, metabolically useful light availability, attenuated phototoxic UV radiation. So this may be a nice little shelter for them. And we also just published this work. To summarize, the histories of the enzymes, they can be amenable to informatic and evolutionary reconstruction. And such reconstructions, in my opinion, are opening entirely new possibilities for the study of early life, including the fine resolution probing of geochemical conditions and stellar properties. And let's go back to our major question. We want to know whether what we see here is a particular case or a general category. And as my friend and colleague Sanjoy likes to put it, exoplanets are one of the, early Earth is one of the best exoplanets you can study. But there's a lot of unknowns, and I know some of you are filling this bingo, so I'll just help you out. Just there's so much we don't know. And study of early life is all hands on deck problem. We need everybody in this endeavor because we cannot find what we're looking for if we don't understand it here. And we owe it to this life on this planet, I think. And with that, I will end. First and foremost, my lab. I'm no longer by myself in this pursuit. It's amazing how many people are excited about this in my group. It's just one of the best. I created a lab that I wanted to be a part of. So this is just really makes me very happy to work with them every day. Our collaborators for different projects and funding comes from NASA, National Science Foundation, John Templeton Foundation, NIH, and also Human Frontiers in Science Young Investigator Grants, as well as University of Arizona and Wisconsin Foundation. And also the Spanish government that is funding one of our postdocs here. And also our cohort. We just started our ICAR about 6 months ago. I am already we've been doing a lot of cool work. I think we created a really great community that is tackling this problem of understanding whether we can integrate inorganic thinking into habitability from very different angles. I'm very grateful to work with them as well. And the names in yellow are presenting here. The asterisks have talks or posters. And finally, we are launching a really cool astrobiology program at University of Wisconsin-Madison. So I hope the students interested in these problems will check it out and join us. With that, I will end.
Thank you very much, Betül. We're close to the bottom of the hour.
Oh, sorry.
But I would invite everyone to stick around who can do without coffee for a few minutes so that we can enjoy a question and answer period. And I'm sure there's going to be plenty of questions. And I think we have somebody who can answer them. Well, first of all, one of our guests last night here is Gideon. Ask the question, please, but speak loudly.
First off, thanks, Betül. It was an amazing talk. So happy to have you. Wesley Swingley from Northern Illinois University. Kind of a philosophical question. Sorry, it's cutting it. A philosophical question. You mentioned that these things aren't deterministic or inevitable. What do you think that is between these singular systems like nitrogenase or oxygenic photosynthesis versus things that are big buckets of stuff like carbon fixation or pigments and things like that?
In terms of the molecular level
Just
impact
why do we have one nitrogenase and 10 different carbon fixation pathways?
That's a very good question. I think maybe what determines the adaptation isn't just the enzyme itself. And I think it goes back to our lack of understanding of how Earth and environment impacted these molecular innovations. So I don't quite have a good answer when it comes to evolutionary biology, because that's not even, I think, a question that many evolutionary biologists are tackling right now. But it's so we have a big question mark as to why there are certain constraints.
All right, thanks.
Can I ask a question from online? So, can you apply this technique to chemosynthetic metabolisms and estimate the likelihood and time for the origin of them? Maybe methanogenesis or iron-reducing bacteria? Can we translate this information to estimation of timing of origin for similar possible mechanisms on other worlds?
Yeah, I think if we have to be very careful. I think when we set up the system, we outlined the first criteria, and I published that in 2017. I'm writing an updated one as to what sort of criteria I think is necessary for reconstruction of ancient metabolisms. When it comes to the timing of origin, I think we are now able to not only reconstruct a single gene, but we can engineer entire communities. So there's nothing that stops us from engineering a community, and maybe that goes back to the Wesley's question, that if we understand how these communities are interacting, we might be able to understand whether there is factors of determinism. So yes, I think it's definitely a possibility, but just be mindful as how you pick your system and be very careful with the design. Happy to help.
Thank you. And those questions were from Sean, Dominical Goldman, and Kendall Lynch.
Oh, hi, Sean.
Next question.
Yeah, hi, Betül. Beautiful talk. Stephen Fried from Johns Hopkins. This is a question coming from a fellow biochemist. I was curious about, so nitrogenases use lots of chaperones to facilitate their biogenesis. So I was curious if you were surprised that the nitrogenase was able to use the extant azotobacter chaperones, and did that map with where the mutations were in the ancestral node, like, you know, where on the surface they were, and did that surprise you or not surprise you?
Yeah, so, so I'm in this point where everything is surprising me and nothing is surprising me at the same time. It's a very quantum state of existence. But we have not done an in-depth network analysis. I think our postdoc Bruno here, he's interested in that exact question. We actually just talked about that in one of our group meetings. So yes, we're definitely interested in understanding at the more larger level, systems level, what might be happening. I think it's fascinating that we're even able to swap these highly constrained systems. So yes, definitely on our bucket list.
I had a fantastic talk. I had a question regarding the rhodopsin. You indicated that the change in frequency optimization of the rhodopsin suggested of that this particular rhodopsin involved in. Could it also indicate a change in the peak output of the sun a billion years ago? And so it's actually reflecting physical something happening in the sun, the surface of the Earth.
Yeah, so what Eddie has done was independent then from our calculations. So he did look at the irradiance profile of the sun itself, independent of, before we integrated and interpreted the phenotype that we've predicted that belongs to these ancestral proteins. And then we sort of mapped them as to this is the environment, and it matched it in the water column where the host organism might be residing. But we haven't done any direct test that focuses on the sun or the star itself, although I think Eddie is definitely interested in looking at the other stars and the possibilities in different times of luminosities.
Do you know which factor was stronger, the local environment or the solar irradiance profile?
So I am very cautious when it comes to interpreting the early environments and what sort of possibilities there might be. We sort of very conservatively propose that perhaps they were either deeper, they were where they could be located in the water column, or maybe there was a biofilm that was covering these organisms. But I want to be very careful since we don't know anything really about those environments. So I won't speculate. Therefore, it may be more likely. I think there's more information about from the astronomy and anyway when it comes to understanding these early models.
Thank you.
Thanks.
Next question, please.
Yeah, your story about nitrogenase was quite inspiring as an outsider to the field. I was really impressed with what you could do with it. Now I'm going to ask you something really unfair. What about Rubisco? How far back do you think that goes, and why?
So Rubisco, this is a very fair question, first of all. So Rubisco is a bit more, obviously, a different type of beast. If you attribute the presence of early carbon fixation records to Rubisco or something like Rubisco, then you can stretch its presence as early as the first reliable data of the carbon isotope biosignature, right? But the simple answer to your question is that nobody knows how old Rubisco is, and we make this assumption that something like it or itself was there when the carbon fixation, biological fixation was emerged. What we've done is to, after we created the phylogeny, we included all forms of Rubisco from today. And we simply wanted to know whether we can use the sequence information of this enzyme to understand first whether we can find signatures of rise of oxygen on this enzyme. Everything that I know about molecular evolution tells me that this enzyme there will be variations, right? But the assumption is that there's some sort of uniformity. And we looked at where the enzyme is binding to oxygen, to carbon dioxide, and in fact found that some parts of the enzyme was evolving more rapidly than the others that corresponded to the oxygen carbon dioxide binding that also coincidentally belonged to the ancestors of cyanobacteria. So we published this in Geobiology, saying we can constrain GOE using the Rubisco sequence, perhaps, by analyzing the sequences itself. We are not timing anything, but we're just saying that there is a signature on this enzyme that is telling you something about what happened on Earth. After we engineered this inside cyanobacteria, we did the isotope studies, and we didn't go as far. We didn't go pre-GOE yet. That's in the works. We think we have a bit more understanding the tool about evolution of this enzyme before we go to that engineering. But as a first attempt, our biosignature reconstruction was in agreement with what the paleobiologists have been thinking about or measuring.
Fantastic. I'm going to have to go to the library, but I have a follow-up question. Carbonic anhydrase is similar to Rubisco in its fractionation ability. In your phylogeny, did you find any relationship between carbonic anhydrase and Rubisco?
Yes, so I actually reconstructed the history of carbonic anhydrase as well, and in the Phil Translate paper, we published it there. Actually, funny you asked that. So carbonic anhydrase is sort of the worst example. So if anyone's wondering, that's how not to do it, because it's a very, as you know, we even have it in our blood, right? So this enzyme is everywhere and in some form. And it is very hard to track it because of the high horizontal gene transfer rate of this enzyme. So we reconstructed it and then thought, actually, you know what? We really should not use this as a probe because it is all over the place and I cannot constrain anything. So I included that as an example of what kind of protein does not fit the bill for deep time reconstruction.
Fantastic. I'm off to the library.
Thank you very much. I believe that we're out of time now. We've been out of time. But please discuss your questions with Betül. I'm sure she would be happy to answer those questions. And with that, I will thank our speaker for a really wonderful talk. Enjoy Wednesday at AbSciCon.
Thank you. Thank you.